Before we begin the webinar, let's take a couple more minutes to allow everyone to settle in. I see people still logging in. That's great. All right, so let's get started. Hello, everyone, and welcome to today's webinar, Unlocking Immunology Data, Exploring ImmPort Database Access Strategies and AI-Ready Datasets. My name is Sanchita Bhattacharya, and I'm the Scientific Outreach Program Lead for ImmPort. It's a pleasure to have you all join us for this insightful session, where we will explore how to efficiently navigate, access, and utilize ImmPort data for research and AI applications. The ImmPort Immunology Database and Analysis Portal is a powerful and comprehensive resource that hosts a wide range of immunology-related data sets, including data from clinical trials, molecular studies, and experimental research. This session is designed to provide a quick guide to effectively search, retrieve, and explore open access immunology datasets, including AI readiness, through a quick walkthrough of the website, tutorials, and ChatGPT demonstration. Before we begin, I would like to cover a few housekeeping items. The presentation time is 50 minutes, followed by 10 minutes for Q&A. Throughout the session, we encourage you to ask questions using the Q&A feature, and we will address as many as possible during the live Q&A segment. And this session is being recorded, and a copy will be made available for reference later. Next slide. So here is a quick outline for the presentations. First, we'll start with the introduction to ImmPort, presented by Kate Hypes, followed by ImmPort Tools and Resources by Reuben Sarwal from ImmPort team. And then Sammi Smith is going to go over the AI-ready data sets and assessments. And then Reuben will do a quick demo of the use case using the ChatGPT and the tutorials on ImmPort website. So with that, we'll start with introduction to ImmPort, and I hand it over to Kate.
Wonderful. Sanchita, thank you so much for that introduction. I realize that as far as our participants on the call today, some may be familiar with ImmPort, but some not. So I'll just give a brief introduction of the ImmPort resource. So ImmPort does stand for the Immunology Database and Analysis Portal. It is a NIAID-funded resource, specifically from the Division of Allergy, Immunology, and Transplantation, really with the mission of preserving our valuable research data and optimizing it for reusability for the future. ImmPort's data model facilitates inter- and intra-study analysis by applying a consistent data model throughout. And ImmPort is focused on immunology-related data, although it does have a very wide range of conditions and data types represented. We'll get to that in a future slide. But as far as ImmPort, the organization of the resource itself, ImmPort is divided into 4 distinct areas that you'll see on the website. First is the private data, and that is for the data submitters, those submitting data through ImmPort, to upload their data and prepare it in preparation for sharing. All data is de-identified, standardized to the best extent possible, and reviewed and curated by our team personally before sharing through periodic data releases that generally happen about once monthly through the ImmPort resource. And so that brings us to the shared data section, and Reuben will be covering this in much more detail today as far as accessing the data and using it. And so there's a variety of tools and resources and ways for you to search and filter through the data as well that Reuben will be covering. ImmPort does have data analysis tools, limited set, but ImmPort does also connect through the NIH cloud available tools as well. So there's opportunities for connection and further analysis on the cloud. And then ImmPort does have a variety of resources for the community in the purple resources area, as well as extensive documentation. So as far as who shares data through ImmPort, so ImmPort shares data from major NIAID-funded programs, as well as external organizations and independent researchers. So you'll see a number of those groups represented on the slide here. ImmPort has been around for over 20 years and is built with fair sharing in mind. ImmPort is a recommended repository from Nature Scientific Data, as well as +1, and has a core trust seal of approval for trustworthy data repositories. All right, and as I mentioned before, Reuben will be covering this in more detail, but this is just a quick introduction to the data browser and the shared data area. This specifically is what we refer to as the cohort discovery tool, which is a kind of a powerful way to be able to filter and search through data sets of interest. As we mentioned before, ImmPort is built on fair data principles with the goal of just making data as findable, accessible, interoperable, and reusable as possible. And ImmPort currently shares data from over 1,100 studies. 175 different diseases are represented within ImmPort, including a wide range of assay types, experimental results, from flow cytometry to ELISA to CyTOF to omics-related data, and much, much more. And the last thing I wanted to point out just briefly in the introduction is ImmPort does track publications that reference ImmPort, whether they be from reuse of ImmPort data, the primary study associated with the deposition of ImmPort data, or just there's been many other publications just about ImmPort in general and the resources available through ImmPort. You can access these publications via the QR code here. The ImmPort team updates this about quarterly. And there are currently over 2,000 publications referencing ImmPort data and resources. And specifically as far as geared towards today's presentation, you can filter by data reuse if you're curious to see how the community is reusing ImmPort data. I think this is my last slide here, but I just wanted to point out that ImmPort has a variety of resources to connect further. If you would like to get in touch with us, there is a help desk email as well. It comes right to our team. We hold office hours the first Thursday of every month from 2 to 3 p.m. Eastern Time. We deliver a monthly newsletter that highlights new data, resources, and features that are available through ImmPort, as well as all of our webinars are archived on the website. So we encourage you to check out past webinars as well if you're interested. And there are a variety of tutorials available, as well as our AI-ready data sets, which we will be hearing in more detail about from Reuben and Sammi today. So I think with that, next up, I know, Reuben, you're going to tell us a bit more about ImmPort tools and resources, and then Sammi, you'll be covering the AI-ready data sets. And Reuben, you'll, again, just go a bit into more detail about a use case and using ChatGPT specifically to analyze ImmPort data. So perhaps let me stop sharing my screen here. And Reuben, I'll let you take over.
Awesome. Thank you so much, Kate. Yeah, so as Kate mentioned, I'm just going to go over first a little bit of this core discovery tool that was mentioned, and kind of show how you can explore the studies that are in here. And then I'm going to show some of the resources, how you can download some of these studies, some of the data, and then also potentially, right, like upload your own data if that was of interest to you. So as I mentioned, this is the core discovery tool. This is one of the greatest resources I feel like that ImmPort has. As you can see up here, you can quickly just see some of the research focuses that are captured with all these studies, right? A lot of vaccine response, immune response, infection response. There's a lot of different conditions or diseases studied here, right? Like influenza, a lot of COVID as of late, and then a lot of other conditions. And as mentioned, right, this is a multimodal database. So there's a lot of different assays that are captured here from like proteomic assays like ELISA, there's flow cytometry, and then there's also some sequencing data that has been captured here. A quick note about that, for a lot of these assays, you'll be able to download the actual result files directly from ImmPort. For some of these sequencing data sets, ImmPort does link out to Geo, as that's still kind of like a main repository for these gene expression data sets. But you are able to kind of find metadata and clinical data associated with these studies in ImmPort, and then also find the actual sequencing data in Geo if necessary. But so you can explore that through here. I'm just going to show right here on the left, you are also able to kind of facet all these studies by these different fields here. So you can see right there's like over 1,000 studies, 174,000 subjects captured here. It's a bit daunting, but there are these nice sort of categories that have been modeled out here, which you can explore. So for instance, if you are interested in like, let's say vaccine response, you can click here. You'll see there's 347 studies that are noted to look at vaccine response. Right, you can see that there are a few that noted that they are both vaccine and immune response, and you can see that in this research focus column here. But really, this is great. You can see all the different 347 vaccine response studies here. You can see the conditions that's associated with this vaccine response study, and then you can also see all the different assays that were run and the numbers for those that were run for this study. And then you can also see the program name. So for instance, if you were interested in vaccine response, right? If you only wanted to look at vaccine response in association with, let's say, like influenza, you see there's still 115 studies there. And then you can even get more granular. If you, for instance, know the exact program that you're interested in, right? I'm going to say, like, let's say HIPC. Right, you can filter and you see there's still 64 studies. It finds it very fast. And then you can scroll through these. I'm gonna just filter this for a second here. Right, and you can scroll through all these different studies that are captured here. There's like all these different pages of studies that you're able to find. I'm just gonna go to an example here quickly. Sorry, over here. And so let's say, right, you were interested in this study, this STY212. You were able to kind of just click into this, and then you get a really nice sort of description of what this study is looking at. Right, you get the sort of more detailed description of sort of the endpoints that were captured. And here you get details of these endpoints. If there's an intervention agent, for instance, like a treatment, that would be captured here. You can also see some of the demographic information captured down there. And then also up here, you can look through these different design tabs to see really specifically what the exclusion-inclusion criteria were. There will be some study protocols that you can explore as well. And then, as I mentioned, right, the different assays that were captured. And so sometimes there will also be studies that will upload data in the study files tab. There's oftentimes a description so you can really see kind of what this data is. So for instance, right, this Excel file, right, apparently contains the CMV associations and these vaccine response data for these subjects here. So that's one way, right, to explore some of the data. If you are, let's say, ready to download this data, you can download that data right here, right? There's a study package option, which is a web option. And then there's also API endpoints, which I'm going to go through in a little bit. So this is sort of a great way if you want to just explore different studies in ImmPort. You can facet by these different categories and explore. You can also just do a simple free text search if you just want to explore that way, and it will free text search all of the fields here for whatever you might be interested in. I do want to now just quickly navigate over to the study registration. So if you were, for instance, interested in actually uploading some of your own data, you would just have to, it's free to become an ImmPort user. You would have to just register as an ImmPort user. Then you can log into this sort of private workspace where you can start to sort of upload some of your data. So I'm just going to navigate to this one quickly and show sort of how that process would look like. So here, right, I've registered this sort of test study. And then there's all these really nice sort of interactive fields that you can go through to start filling out your study information. So for instance, right, there's, as I showed, sort of when you click into a study, there's a detailed description you can put in. There are these endpoints, right, as I mentioned. You can even put in like what sort of hypothesis you were testing. And then these other sort of like clinical demographic information that's important. And then as you navigate through, right, you can go through these different categories, right? You can note the different arms or, right, sort of cohorts that were studied, the inclusion exclusion criteria, and all these different fields that are important, right? Upload some protocols if you need. And as I mentioned, you can upload study files that are sort of associated with the data that you want to upload. So this is a really nice sort of user-friendly way ImmPort makes it for not only, right, finding data, downloading data, but also, right, uploading your own data. And so this is a private sort of workspace. And so you, as the uploader, are the only one that can see this. And then when you are ready to make this data public, the ImmPort team pushes that out to sort of the public space for everyone to see. And so lastly, I just wanted to navigate over here to the resources page, just to mention, right, that ImmPort also has a lot of great documentation if you ever get lost in terms of trying to find data, download data, upload data. So for instance, right, they have a great API documentation if you want to, if you feel more comfortable downloading data that way. There are examples, right? You just, in the terminal, you can just put in your username and password. And then there are great examples for how you would pull, for instance, if you are interested in ELISA data, right, you can use this endpoint ELISA and you can pull all the ELISA data, right, for multiple studies of interest at once. Which is great. And then it's say if there are other endpoints you are interested in, let's say other assays, you can navigate to here and you can see what other, what these endpoints are if you want to pull these other assay data. So with that, I am just going to hand it over to Sammi, who's going to talk a little bit more about the resources that are found here on the resource page, specifically the AI-ready datasets and the assessment.
Thanks, Reuben. All right, just sharing my screen here. All right, so the next topic, as Reuben just said, is we're going to talk a little bit about the ImmPort AI-ready datasets. So right now you're seeing we're back at the ImmPort homepage, and I'll just show you guys how to navigate to this resource that I'm going to be talking about. So from the ImmPort homepage, you want to navigate to the banner up along the top and click the purple resources section. So at the top here, we have many different resources for particular programs that deposit data into ImmPort. And then if you scroll down a little bit further, we have this additional resources section. And what we're going to highlight today is this first one here, the NAIR AI Ready Datasets resource. So if we click on that tile, it's going to take us to the page. This is a collaborative effort between ImmPort and the National Artificial Intelligence Research Resource, or NAIR, in their pilot program. This is an initiative that has aimed to support AI research, and one of the ways it does that is by providing datasets that are structured and optimized for AI applications across lots of different fields. So in our particular case, the ImmPort team provided datasets for immunology and infectious disease. So to provide a little bit of background about the NAIR pilot program, it's a federal initiative that started a couple of years ago and it's still ongoing. It is a joint effort led by the National Science Foundation in collaboration with other US federal agencies. And it's designed to connect US researchers and educators with computational power, data science tools, training resources, really all the kinds of stuff that you would need to advance artificial intelligence research. They've collaborated across different federal agencies with government and non-government partners. And the goal of the pilot really is to lay the groundwork for more comprehensive AI research infrastructure on a national level. So as part of this effort, last year we began our partnership with NAIR, and we're continuing the partnership this year. We've already made available 2 AI-ready datasets, and we're currently working on preparing a third one that will be shared later this year. So if you scroll a little bit further down on the page, we have a nice summary table that gives you more information about the available datasets and allows you to download them and use them in your research. The 2 datasets that we're going to talk about today are 10K immunomes and the COVID-19 dataset. These datasets are designed to contribute immunology data to the NAIR research community. So they're both available and hosted on this page. You can simply click the download button for either of them, and a download will be triggered as a zip package to your local machine. We also give this description here that gives a little bit more information about what type of data is contained, so you can decide if it's something that you're interested in exploring further. We also provide something we call our AI readiness assessment for each dataset, which I'll explain a little bit more about in a bit, as well as information on how to cite a particular dataset, including a DOI associated with each individual dataset. So I'm going to talk briefly about the content with each of these 2 datasets. The first one is 10K immunomes, which you can get by clicking this download button here. This one is a dataset that compiles immune system profiles from over 10,000 control subjects, and that data was derived from 83 studies that are hosted within ImmPort. I've already clicked on the download button to download it to my local machine and unzipped it, so you guys don't have to wait for that, but I'll show you guys what that data package looks like for this one. So dragging this across to the screen here, you can see that it opens up where at the top level we have a README file that explains a little bit more about the structure of the files, and then we have things organized into subfolders based on the particular subset of data that you're interested in. It has a large variety of different data types. So we have some flow cytometry data, some CyTOF, mRNA expression, etc. And it's really designed to serve as a reference resource for human immunology research. So I'll go ahead and drag this off the screen now. The intent of this data set is to serve as a set of healthy controls, because when we're trying to understand pathology within the immune system, it's oftentimes really helpful to compare those different experimental and lab values that you're getting with what expected values would be for a healthy individual. In the description column, we also have some additional pieces of information about the data set. So we have a publication here associated with the data set that you can read. We also have a link where you can browse the ImmPort studies in that cohort discovery tool that Reuben was just talking about. And then we can also link out to the source code available on GitHub or Docker. So now let's turn our attention to the second data set. This is the COVID-19 data set. This one is a harmonized collection of curated COVID-19 studies. And it is a conglomeration of 18 ImmPort studies. The dataset includes key immunological data types that help us to understand COVID-19, so flow cytometry, ELISA, neutralization titers, etc. And it's designed to facilitate meta-analysis for AI-driven insights. This dataset was compiled to support the SeroNet program in particular, as well as other communities who are researching COVID-19. And again, this one has a similar link where you can browse the ImmPort studies in shared data. So I'll show you guys what that one looks like. Basically, it opens up the cohort discovery tool already pre-filtered by the SDY numbers that are contained within that data set. So if you want to scroll down and find more information about each of these data sets in question, you can see the full data set where it came from. All right, so now that you have a good idea about the content of the available AI-ready datasets, I'm going to turn our attention to explaining the AI readiness assessment section. So let's scroll down to that. So as part of making these data sets available, the ImmPort team really wanted to ensure that these data sets met standards for AI applications. And so we conducted a search for existing resources out there on assessing AI readiness of a data set. And we found some resources out there, but really not a whole lot. So what we ended up doing as part of this project was utilizing a checklist that was published by a research group in the earth sciences field as somewhat of a starting point, and then we modified that checklist to be more relevant and specific for immunology data, since that's the space in which ImmPort operates. We also added a suggested scoring system to the assessment as well. So the questions on the assessment are really designed to encompass 4 broad areas of what we're referring to as this concept of AI readiness. We have data quality, data documentation, data access, and data preparation. In addition to our team using this assessment to help us prepare the 2 data sets I just described, we also make it available as a blank template version, so researchers can utilize it to assess their own data sets if they're interested in sharing them. So if you click on the graphic here or any of the other links we have within the text, it will open up the AI readiness assessment in Google Sheets format. And you can go ahead and go up to File and select Make a Copy if you're interested in assessing a data set that you already have available. So this first tab here that it opens up to is the Overview and Instructions tab. And this part just kind of explains the intent behind the assessment, how do you use it, and what is our suggested scoring system. And then if you navigate to the second tab, this is the assessment itself. So we have all sorts of questions for you to fill out and a column for different definitions that we link out to explain a little bit more about any vocabulary words within the questions. And then we also have a section for notes, and then I need to zoom out just a little bit here, and then you can see the scoring section on the far right. So if you scroll down, you can see each of the 4 sections that I touched on. After you answer all of the questions in the assessment, you're going to zoom out and assess each section as a whole and take all of those answers into account and assign the section a score from 0 to 4. And then once you've assigned a score for each of the 4 sections, down at the bottom, an overall score will automatically be calculated for you as a simple average. So something that we're really trying to stress here with this AI readiness assessment is it's not meant to be an exhaustive list of requirements that you have to meet, but instead it's designed to be something that helps you identify areas you could potentially improve in your data set, and primarily to make sure that you haven't overlooked something that might actually be really beneficial for your data set in order to make it more usable for downstream AI applications. So please don't feel intimidated by the checklist or view it as a barrier to sharing a data set. It really is just designed to be an exploration around your data to assess where you're at and potentially find ways to improve it from an AI usage perspective. It also serves as a really helpful piece of documentation or metadata, data about your data, if you will, that you can share when you share a given data set that will help downstream researchers understand the ins and outs of your data. And that's exactly why we chose to share our assessment along with each of these 2 AI data sets up in the table at the top. So back to the resource page. At the bottom, we just have some additional information for you about NAIR. You can click on either of these 2 images to take you to the nairpilot.org homepage or the NAIR page on the National Science Foundation website. Both of those give you more information about NAIR in general, what the program goals are, and you can keep updated on any future developments for the pilot. And finally, we have suggested citations for this page itself, as well as a reminder about the citations for the data sets specifically. So to wrap up this AI-ready datasets topic today, whether you are an AI researcher, an immunologist, or a data scientist who's just interested in immunology, these resources do offer valuable high-quality datasets to support AI-driven discoveries within this field, and we're excited to make them available to our users. Now I will turn it back to Reuben.
Awesome. Thank you so much, Sammy. Yeah, so as Sammy mentioned, I'm gonna now go over a little bit of how ImmPort is providing resources to sort of help analyze some of these data. And then I'm gonna touch a little bit on how these novel LLMs like GPT can actually help analyze some of this data as well for like a novel researcher. So for instance, like I ended sort of here on the CDT where, you know, you can explore these different studies, right? I had filtered by vaccine response, specifically influenza, and then just one more, I was interested, let's say, in these HIPC studies. So let's say I find this SDY212 that I'm interested in looking at. Right, as I mentioned, right, you can sort of see the endpoints captured here. You can also see the different sort of like arms that are looked at. Right, there's like some sex information. So let's say I want to actually start analyzing some of this data, but you know, I'm a little bit novice to sort of coding or analysis in that way. ImmPort has, right, these great resources. As I mentioned on this resource page, there's a tutorials tab which you can explore. And then there are these great resources for just providing examples of how to start exploring data and doing some basic statistical analyses. So here's that 212, for instance, that I just showed. And then here are some example codes in R as well as Python. So if I click into this one, to the HTML, right, we get this really nice R markdown, which kind of goes through sort of how you would begin potentially sort of exploring some of this data. Right, you can load in the packages of interest and the libraries, right, and then start sort of just pulling the data from ImmPort. Right, these are these like text files that if you download, as I showed earlier from that download tab, you get these like text files which you can easily sort of read in. And then there's great examples of starting to like explore the data, right, and creating some nice visualizations. Just doing some descriptive statistics between, right, these ARM names, which is old and young, by the sex information. There's also some race information which we can plot. And then you can actually start looking at, right, some of the assay results, right, these titer results. And again, this is just a nice sort of tutorial, nothing too complicated going on, but just a nice way to start exploring and visualizing the data. And then, right, there are even some statistical analyses that are done down here. If you want to see, right, are there differences between these old and young groups by these different viral strains? So ImmPort has these great resources, right? This was for this HAI data, right? But there's also some great tutorials if you're interested in flow cytometry data, these other sort of lab test data, and then even some more sort of deep learning tutorials for some more advanced users. So I'm going to switch gears quickly just to now talk about how you can utilize some of these LLMs, such as GPT, to actually do some of these very similar analyses that I just showed that were done here in the tutorial. I'm going to switch gears here. So I mean, the main step, right, as I mentioned, right, is kind of seeing what data you want to first explore and look at. As I mentioned, right, as I just showed, right, there's the CDT. You can filter for all of these, research focus and conditions to find the studies of interest. Right, you can also filter by species, right? ImmPort does allow for animal studies as well. But if you're just interested, right, in human subjects, you can filter for that. And so, for instance, I'm going to take, for this example, data from the study SDY396, which is another vaccine response study. Here they're looking at sort of these different vaccines, this trivalent influenza vaccine and this live attenuated vaccine. And then there's also these different analytes that are captured in response to these different vaccines, basically these immunoglobulins, the IgG, IgM, and IgA. And then these were taken over multiple time points to see how, right, the immune response changes or wanes over time. Quickly, just a little background, why did we choose these studies? For instance, just for, purposes sort of also of this tutorial, right? These were chosen because these were really clean studies. There wasn't much like missing data or cleanup that was sort of needed before doing analysis. But, of course, as many people know, right, data cleanup is always a part of doing analysis. So that shouldn't really be something to dissuade you, but something to note if you are going to try and use some of these LLMs. Right, GPT can help you use data, or clean data rather. But it's always good to know sort of what your data looks like before going in. So you know sort of what the LLM is doing. But as I mentioned, right, GPT is great for sort of just doing these exploratory analyses that I showed. So with like one prompt, right, I can upload the data. And then I could say, I'd like to just sort of look at these basic distributions between the groups, right? You can specify the arm name column, since I know that is a name of a column in there. Right, but you can also, right, treat it more like a person and say, I'm interested in looking between the different vaccines, right? And the LLM is pretty good at knowing, okay, like, this TIV is probably a vaccine name, so I'm going to look at that arm name column. But it is always good to also specify if you're able to. But yeah, so just with one prompt, right, you were able to get these really nice visualizations, right? Very similar to what I showed in the ImmPort tutorial. But you can look at sort of the distributions by these different vaccine types, by the race, by sex. And then also, as I mentioned, right, GPT can also run these basic statistical tests. So similarly, as I showed in the tutorial, right, if you just want to look at between these groups, right, is there any significant difference? GPT can do that very easily, right? You can specify also, again, just with one prompt, I want to look at this specific analyte and see how this changes over time in this group. You can specify the exact statistical tests like non-parametric, parametric. And right, I did not prompt it to create box plots. It knows that's probably, right, the most logical way to plot this kind of data. And so, yeah, so you get a nice plot, you get an over result, and then you also get, GPT provides like a little interpretation for you also as well. So that's great. And so this was done again, right? One prompt, I just said, I want to look at this analyte. And now, right, with an even simpler prompt, since it knows that worked well, that's the prompt I wanted, and the output was good, I say now just do the exact same thing, but looking at these other analytes. And just, right, in a kind of like a minute or so, right, it plots all these and also gives me the p-values. And then, right, I can do this similarly for race and by sex. So yeah, so GPT is great. I mean, yeah, it's able to do a lot of this stuff. Now with sort of the LLMs like kind of advancing, I would say like by the day at this point. There's also now these new functionalities where GPT is sort of able to actually explore the web and download stuff from websites when applicable. So for instance, here I'm showing, right, since there was some interest also in geo, I'm showing, right, GPT can also create sort of pipelines to actually pull data from geo. So here I gave it the exact GSC number, for instance, and I said, can you give me the full pipeline in R to kind of pull this data from geo and just do some basic differential expression analysis using DSeq. And again, right, in one prompt, it gives me a nice full pipeline, which I just copy. If you can see my cursor here, right, I just copied into my R Studio and ran it, and it was able to, yeah, it worked. So yeah, so GPT, right, these LLMs are amazing. They can do a lot of interesting things, and I really do encourage everyone here to kind of explore what they can do. As I mentioned, they can do sort of like hypothesis generation. They can interpret your results. They can interpret figures as well. There's sort of these like new tasks that we in the lab have been exploring. For instance, we use GPT to do cell type deconvolution, which is pretty exciting. But now we're also sort of using these LLMs to actually create machine learning models. So this is sort of a work in progress that you can see down here, where we utilize these different LLMs. We uploaded these different data sets. And just as a note, actually, so this March of Dimes, this team actually used the AI ready assessment that Sammi just presented on to sort of score this Dream Challenge data set. And now we're utilizing it for this LLM to sort of bring it full circle. But yeah, you're able with prompts, right, to actually create models and also like optimize your existing models if possible. So GPT can do a lot. It's a really great tool. I will say though, just, there's always limitations, things to consider, right? Always important to check GPT's work. Always check your own work, of course, but always check GPT's work. By nature, these LLMs will always give some sort of output, and this can, sometimes not be correct, obviously. For instance, just as an example, right, if you are saying like, I want to look at a certain variable between groups, if, for instance, in a certain data set, maybe that variable is not there, sometimes GPT will just like impute random data or like force that variable. And like really it'll be a garbage output, right? Because that variable does not exist. So it's always important to know what your data looks like going in, and also like inspect the code as I showed here. Kind of inspect the code that it's running and actually run it yourself if possible to see that, the output is actually making sense. And yeah, prompting is going to be kind of the most important thing, I think, moving forward. I think it's also always good to sort of look up, how best to prompt these LLMs. There are these things like system prompts you can use where at the very start of your session, you can say like, I want you to pretend you're a, geneticist or like, you're an expert in coding. And these sort of system prompts can sometimes help sort of gear the LLM to a more specific task that you are interested in. But yeah, so this is, I think, my last slide. And I think now we're going to take some questions. I'm just going to leave this up here for now. And yeah, take any questions whenever.
Yeah, so I could see there are a lot of excitement around AI readiness and GPT analysis. So we'll take some of the questions live. Let me start with the first one. What are some of the challenges you have encountered so far in making data sets AI ready? And how have these influenced the strategies you have or will implement as you work to make more data sets AI ready? Probably Sammi and Reuben, you guys can answer this.
Yeah, thanks, Sanchita. I can definitely take that one. So yeah, there's certainly challenges that we've run into with making data sets AI ready. I would say one of the primary challenges or things that we really tried to focus on was the understanding that the metadata for a data set is really important. So if you're trying to pass in, just a spreadsheet or a CSV of different values to an AI model, and it doesn't really have the context of where those data values came from or, an intelligent understanding of what the header values mean, it's going to have a hard time deciphering that data or making valid analysis conclusions. But if you can provide a little bit additional context when you're feeding that data into the models, you're going to have a lot more success. So just recognizing the importance of metadata and making sure that when we publish data, that metadata is published along with it. Reuben, anything to add about any other challenges or anything you'd like to add?
No, I think you, covered it pretty well. Yeah.
Okay, I think then we can go to the next question. Probably that's more targeted to Reuben. Can GPT run joint tests for such longitudinal data over time, and which would have greater power to detect distinct time trends?
Yeah, so I will say, yeah, so GPT can, I guess, a slight caveat. So GPT has like what I showed earlier in the tutorial. So GPT can run analyses right within the session and then outputs. It can also create some outputs right within that session, like those box plots and stuff I showed. For sort of like these more complex analyses, right, like a longitudinal analysis or something, there, right, you can prompt GPT to give you like the code to run that. However, like within GPT itself, oftentimes I've found that if with those like longer analyses, the session will sort of time out and it won't be able to actually create the output itself. But that's where I think it's useful to ask, can you give me like the code, for instance, of how to do that myself? And then you can just copy that and run it in your own like R, Python, or Jupyter Notebook.
Yeah, I think there is another follow-up question. Has anyone used GPT to detect heterogeneity among as of yet undiscovered groups, like clustering of by differing trajectories over time?
I don't know if GPT has been used for that specific task. However, I do know, like within Sanchita in our lab, there is a lot of discussion, right? Using GPT as sort of a gene ontology, sort of resource, right? As some of you might know, right? Gene ontology analysis right now is sort of limited to right, these ontological terms and like these reference data sets that have already been sort of established. But GPT is not necessarily limited by, right, these rigid sort of like reference data sets and ontological terms. So there, right, it's very interesting to see, right, if you have a differentially expressed gene list, to say like, is there anything interesting in this list related to my disease of interest that's maybe not in literature right now, or maybe it's not in gene ontology as of now. Yeah.
Yeah, maybe I can also add that we are still in the exploratory phase where we are taking import data sets, analyzing it, and validation is very important. We want to make sure that, not fully relying on ChatGPT, but also going back and checking through all the statistical methods. Are these, the way to analyze the data, making sure, and validation is very, I would like to stress enough on that validation is very important. It gives you a kind of a guidance, give you ideas, but we really want to make sure that whatever the code and the types of data analysis methods being suggested is working and getting validated. All right, so since we have more time, so that's good. We have another over 10 minutes. I'll take the next one. So Reuben, are you using the advanced ChatGPT for your analysis?
Yes. So actually, what I showed in earlier about just exploring the data, that was using GPT-4. But so for actually, maybe I can go back here. Sorry, quickly. For this work that we're doing here, yeah, we're using the more advanced GPT-4 model. So like the O3 mini high, which is supposed to be specifically geared towards logic and reasoning and coding. And then yeah, these other sort of non-OpenAI LLMs as well.
The next one is really, Reuben, you can provide the resources. Those are available for prompts on import data sets. I know we have workshops that we have been organizing to do some of the exploratory analysis in ChatGPT, and later in the year we're going to have a AI-based symposium that will be organized by ImmPort, where we have about a 2 to 3 hours of working session, hands-on session, where we teach the attendees how to develop the prompts, do some analysis using import data sets. Per se, we don't have it's not a resource for more, it's a hands-on workshop at the moment.
Right. And then just related to that, OpenAI, and I'm sure these other providers, they have great also sort of resources for how, sort of best to structure a prompt where you list your goal first and then sort of the problem area and then what you want your output to be. So there are also resources like provided by the people who make the LLMs on how best to structure prompts. But yeah, we have like some materials which, yeah, I think as Sanchita mentioned, that we can maybe start to also like publish as like good resources and prompts for exploring specifically immunology data in ImmPort.
And I can add on to that a little bit as well. So we oftentimes find that prompts are very specific to a certain model as far as what works well and what doesn't work as well. So if you're using models from OpenAI versus models from, Grok or some of the other providers out there, they may not have the same level of success with the same prompt. So it's really kind of a matter of trial and error sometimes and understanding what structures work well for different models and different contexts. And even within one provider, like something that's going to work well for a GPT style model from OpenAI is probably not going to be the ideal structure for one of their reasoning models that are part of the O series of models. So it's really just trying to keep up with all the best practices out there from these rapidly changing models.
All right, let's go to the next question. Is open source contribution allowed for making import data sets AI ready?
I can take that one, Sanchita. So that is actually something that we had considered, maybe adding a community curated section to our AI ready datasets page. Right now, the AI ready datasets are they're all prepared by members of the ImmPort team and published there. But it would be really nice if we could have other researchers provide datasets that we could list on the page as well. What we've done, it's closest to that so far, is we actually have partners with the March of Dimes project who were really interested in this concept of AI readiness and AI ready data sets. And so what they did is they built out their own AI ready data set and hosted it on their March of Dimes page. And they followed kind of the same structure that we used within ImmPort. They even used our assessment template to assess their own data. So if that's something you're interested in, feel free to reach out to us.
All right, there's another question. Did you find something AI can detect but manually analysis cannot?
I can take this. So, I mean, this is the work in progress that we are still submitting. But yeah, we did find specifically right for this dream challenge, which actually utilized this like from the March of Dime folk. They utilize a crowdsourcing sort of platform to have a bunch of different like teams try and build the best model to predict early preterm birth. And we are seeing that some of the models that we built kind of using LLMs, some of those have outperformed the teams that did the crowdsourcing challenge. So there definitely is some very cool opportunities for advancement and sort of, yeah, beyond what I guess, right, people can do by themselves. But as Sanchita mentioned, right, there's still lots of benchmarking work kind of needed to be done before taking it there.
Great, I think we answered most of them, right? All right, let's see if we have any left. Okay, so I think it was a great presentation, and thanks to Reuben, Sammi, and Kate. And also I want to call out the other team members, those who have helped me to organize this webinar, Emma Kendry, Zulinel, Trila Flores, Matt Straub, Ruth Montero, and Elizabeth Thompson. And as you can see, we continuously develop new data, like new types of analysis or resources or tutorials, so the community can get to keep community engaged with data reuse, promoting data reuse from these public data sets. If you are further interested, please stay connected to ImmPort. There are many different ways you can, like ImmPort office hours are hosted every first Thursday of the month. And then we have ImmPort newsletters. Feel free, here are the QR codes, so you could scan and register to the ImmPort newsletters. And then the ImmPort webinar archives, as soon as we are done here, we will post the recording on a YouTube format, so you can go ahead and try out other webinars that we have conducted in the past, or this one. And then ImmPort tutorials. So we have ImmPort tutorials on the website, so you can, and Reuben showed one of them, but we have other tutorials as well. And then ImmPort AI ready data sets. We will in future, we will be publishing other AI ready data sets. There are some very, very cool data sets in ImmPort, which we would like to make it AI ready. And so with that, I think we are finishing 5 minutes early. We're going to close the session, just making sure if there are any other for the Q&A. So I think we are good. So all the questions got answered. Still, you have 2 minutes if you want to ask any other questions to the team. All right, then we can give you back 4 minutes and see you in the next webinar. Thank you.
Thank you.